Being able to measure single-cell RNA-seq on a 3D coordinate system would better improve the current knowledge of spatial developmental biology, especially when this contains only single-cell RNA-seq information from BAR-seq2. Now, BAR-seq2 is able to measure the 3D coordinates of single cells jointly with single-cell RNA. In this analysis, those 3D coordinates are removed, hidden, and not used in the analysis. And so we're only using single-cell RNA in conjunction with spatial transcriptomic information. So the spatial transcriptomic information contains both pixels and pixels as images and gene expression. And what I showed below in the 4 slides are spatial transcriptomic images that are predicted from the actual observations. Now, the motivation for this is to grow better organoids, which can eventually form better organs and tissues. And the main aim of this research focus is to create organs that are able to repair and regenerate. So being able to map single cells to spatial structures definitely improves our interpretation of spatial developmental biology, and it would be able to map the genes and how they progress and evolve spatially under certain cues and directions in order to develop the organ tissue. So what I mean by this is, for example, the gene called SIX2, which is a key gene in kidney development. And this was only uncovered through better understanding the spatial biology of the kidney. I show 2 quotes from the 2 papers below, and you can read more into it, and I provide the titles and the DOIs in the images. So the datasets are 1 from spatial transcriptomic measured from the Allen Brain Institute, provided in the GitHub link, and this is for the competition called CAST, also from the Allen Brain Institute, and they provide, I believe, BAR-seq2, single-cell RNA-seq measurements, but they remove 3D coordinate systems that BAR-seq2 can extract from the single cell of the mouse brain. So with the data, I split into training and test, where the training contains the spatial transcriptomic histology of both the pixels and images, and I also look at the test set, which contains only single-cell RNA-seq. So I use the images, especially the pixels, as a training. So I attempt to predict the pixels of the histology using only the gene expression. And with the gene expression, I then build a model that can predict the spatial coordinates of the single-cell RNA-seq. So in the bottom rows, I show the expression of a gene called SLC24A2. And later, I'll show how both the observed gene expression of the spatial histology aligns with the predicted gene expression of the spatial coordinates for the single-cell RNA-seq. Now, what I show here are 6 images. 3 of the images on the top row are the same as the 3 images on the bottom row. So these images are predicted spatial transcriptomic tissue based on a 3D coordinate distribution or prediction of the locations of the single cells. Now, you can see in the top row that when you align or draw a line along the axis of which the tissue is most variable or has the most range over, and then align them based on the angle of the direction, you can build a coordinate system that looks like a 3D prism, as I show in the bottom figure on the right. So you can imagine that each of these three columns representing three different perspectives of the coordinate system can be imagined as orthogonal systems, or essentially the planes X, Y, and Z. Now, in the bottom image, I show, for the bottom rows, to be more specific, I draw lines along where I believe the cell colors, especially the gradients, are most differentiated. And so that will define the cell populations, colonies, and types in the predicted spatial coordinates of the single-cell RNA-seq. And soon I'll show the gene expression over which these areas cover, and you'll soon see that these regions, more or less, are defined by cell types that express different genes in a spatially different way. But first, before that, I'd like to look at the spatial transdynamic information again. So in the top row, I show only spatial transdynamic histology of the observed training set. In the bottom two rows, I show the predicted Z or Z-axis and the predicted Y-axis, where I assume the X-axis is the top row given by the observed measurement. So for example, if we take the last column and we look at the middle image, as I show with my mouse cursor, if you rotate this 90 degrees anti-clockwise and then align it on top of the image, you can see that each section here is green, followed by white, followed by pink, and followed by several regions of gray and dark green. Now remember that of green, white, pink, gray, and then if we look at the middle image here, we'll see that it's almost green, white, pink, gray, and that shows that this axis along the cross-sectional tissue of the mouse brain is captured by the cell shown here. And this image is of predicted cells along the y-axis. Yes, the predicted. Now, I also look at the z-axis, that goes down along tissue here. So just remember, when we go down, we have pinkish white, followed by white, and then followed by green or gray. So as I say again, if we look at the bottom image, we have white, followed by pink, followed by white, followed by greenish gray. And that's what I mean by being able to recapitulate a 3D coordinate system of the spatial transdynamic tissue. And from this image, we can definitely see that the three orthogonal axes are being represented by all the images and the information that the model is capturing. So these next set of slides will show gene expression spatially arranged along the cells of the predicted coordinates. Now, what I will do next is show genes expressed in this circle, after which I'll show genes expressed in this circle, and so on and so forth, and move the circle around the spatially arranged single cells, such that they cover the whole predicted spatial transdynamic tissue from single cell RNA-seq alone, where the bar-seq 3D coordinate system is not given at all in this data analysis. So I'll slowly go over these slides next. essentially what I show are the real or observed gene expression only on the far left column of the histology tissue, the predicted Y and Z axis of the spatial transcriptomic histology tissue. And that is what we see on the middle left, the gene expression of the spatial transcriptomic and the distribution of it. And then on the right, we have the predicted spatially arranged single cell RNA-seq on the top, where the colors are the predicted pixels or images of the spatial transcriptomic. And the circles essentially highlight the gene expression of the cells that we predict. Sorry, I'll go back to the first slide. And so essentially the cells predict the locations where the gene expression is most likely to be occurring spatially in such a way that it distributes cells based on gene expression, spatial gene expression. And that's what we see in the middle row on the right. And I just copy and paste this from the previous slide so that we can always have a reference of what we are looking at in terms of a 3D coordinate system. And this gene is of C1QL3. And I'm not a mouse brain expert, but I believe, or I can see, that the gene expression is spatially coordinated. And I'll slowly go over each slide. And there are about 10 to 20 genes in total that I show. But you'll see that the circle moves over the tissue slowly in a orderly fashion. And I'll shift over the slides now. I'll give about 10 seconds in each slide. Now, I hope that was insightful and revealed that this model can represent the spatial information of single cells when given spatial transcriptomic information. Now, the question is how is this model created and learned? Well, essentially, I use a loss function for a neural network where I put in about 6 parameters that define well, sorry, not parameters, but 6 components that create the loss function. So what I noticed is that a loss function can be almost representative of anything. And what I propose is that each component ask a specific question that defines the spatial biology. So rather than optimizing for a certain metric like Euclidean distance or gene expression correlation, I specifically ask questions and attempt to model the metrics or information to answer these questions in a given loss function. And the questions are essentially about cell distributions, cell locations, the gene and pixel space, how well the predicted pixel images are to the local predicted images, as well as the global images. And the last one I find is interesting is how well the movement of cells that are updated iteratively, how well does that fit the idea of Waddington's marble analogy, where Waddington's marble analogy depicts the cells moving around on a differentiation landscape, where at the top of this surface, like a slide, we have the stem cell sliding down into different roots that are more differentiated and mature cell types. Now, I want to go into the neural network, and as we can see here, I have 3 parameters, essentially 3 different linear layers, and that's about it. Or that is it in terms of the neural network. Now, what each of these 3 layers represent, I'll go through below and in the next few slides. But essentially, I have 3 called beta, GEX, and PIX, or PIX. So the beta parameter essentially learns on the genes, or what I call the k-nearest neighbor gene convolutions, to predict the coordinates of the spatially aligned or spatially distributed single cells. Now, what GEX and PIX do is essentially map the gene or images to a shared space. And from the shared space can share information such that we can better transform or predict what is going on and learn more using the model. So what is the KNN gene convolution and how are these coordinates predicted? Well, I'll first look at the images on the right. Essentially, each cell is represented by an intensity spot or dot on the right of the top right of the main image. In the bottom right, I essentially create a KNN grid where each square represents a convolution of the spatially arranged cells. Now you can imagine each gene has its own image as represented in the top. And so if this grid is of 1,000 by 1,000 units, then the G channels or gene channels essentially would distribute this information over those G genes, and essentially creating a sort of image of 1,000 by 1,000 by G. Now, I also apply a spatial diffusion kernel, like a Gaussian kernel, on top of this for each gene, and not across genes, just for within each gene channel. And this essentially creates a continuous nearest neighbor diffusion of information of that gene. So instead of a discrete KNN nearest neighbor graph or network, I have a more continuous space over which I can overlay spatial information on. Now, I then take convolutions, as I mentioned before, and these convolutions are quite small. And then from these convolutions, I put this through a neural network to predict where the cell is next likely to occur. After which I iterate this over this process, and essentially I update a sort of walking process, or a process over which the cell moves to the next likely location based on the gene convolution, or KNN gene convolution. And so this is very similar to Waddington's marble analogy that describes the differentiation landscape. Instead, here we have a spatial landscape, a snapshot or window in time where the genes are most likely to occur based on the histology slide. Now, next, I'll talk about a shared space. So what is a shared space? Essentially, if we have two modalities, y and x, a shared space would be able to transform x to y and y to x using a certain function or its inverse. And this essentially builds a bridge between those two modalities. For example, histology and transcripts. You can build a bridge or transform or shared space between them. Or you can look at epigenetics, or even from genetics to proteomics along the central dogma of biology. Now, what can you do with a shared space? As you can see, before the images were all the same, and now they're transforming into different images. And you can see that these three images represent cells that are changing continuously over time, back and forth from the same states, as we see here, to a more differentiated state, as we see now. So what does this mean, or what can it be illustrated as? If we think of Waddington's analogy of the marbles, we can say that the state here is similar to the stem cell, and the state here is the more mature differentiated state. And now I only loop back and forth to illustrate the idea of Waddington's marble analogy. And I hope that was insightful and enlightening. And so I'd like to wrap it up and provide the GitHub link. So it's github.com/ask, the word ask, to question, explain, to describe, ask, explain, followed by the text scRNA maps, where there's an underscore between scRNA and maps. And that's pretty much it. So I'd like to thank these people for inspiring this work, especially Faye, who I have been able to discuss this regularly with, as well as Alan. And I'd also like to thank Jeffrey McLaughlin and Cynthia Boroli, who have been instrumental in providing the direction for this work in terms of setting up the required knowledge and information and education in terms of deep Gaussian mixture models. And I really do think that deep Gaussian mixture models are a great model and that they should be discussed further. And so I like to end it there and thank the people who have watched this video.